We're going to talk a little bit today about, like Susan mentioned, what I've been doing in her lab in collaboration with many others over the last several years. And I've titled my presentation, Building a Better Mousetrap. And this was the most effective cartoon I could find on the internet that illustrated what I meant by building a better mousetrap. And we'll talk a little bit later about what I think that means towards the end of the presentation. So to start out, we'll have a brief outline, which will include an introduction to rice and an introduction to Ionomics. We will go over some available tools that I've had access to over the course of my time at Cornell. And we're going to talk a little bit about how I've been able to use those tools in order to answer relevant questions. And if we have time, which we probably won't, we'll talk some about the future work and where we want to go and some of the things that we can do with the analysis that we've done. So I love maps. Maps are really fun. And this map in particular is really fun. Okay, so this map comes from National Geographic's 7 Billion project. And this map strikes a chord with me because it's the brightness here is correlated with population density and the color is correlated with income. Okay? And then I've overlaid on top of this map rice as a percentage of caloric intake for many of the people that live and survive on rice. And it doesn't take much to learn that the poorest parts of the world depend very heavily on rice for their income. Bangladesh, right here. Look how densely populated Bangladesh is. Does anybody know off the top of your head what the population of Bangladesh is? Ronnie?
140.
140 million, says Ronnie Kaufman. All right, 154 million was the most recent statistic I was able to find on the internet. Okay? And they get 76% of their dietary caloric intake comes from rice. So it's a really rewarding crop to work on. And rice and I, after my defection from the maize program, have been falling in love for the last 3 years. And I'm really grateful to be able to continue that courtship even after I leave Cornell. So in addition to its importance as a major staple food crop, rice is also really cool for lots of genetic and phenotypic reasons. So for those of you who are unfamiliar, the graduate students on Friday did a very good job, Jen and Juan David, illustrating this concept, but we'll do it again today, that rice is deeply subdivided genetically. Okay? So the prevailing theory is that domesticated rice actually originates from independent domestication events from a pre-differentiated wild ancestor. This creates genetic subdivision in domesticated rice in such a way that we have the indica clade here, and then we have the japonica clade here in blue, and those are further subdivided into the indica and Aush subpopulations, and then japonica is divided into tropical, temperate, and the aromatic varieties of rice. So that'll be important to remember. If you're not familiar with that, we're going to be referring back to it quite often. But the other thing that's really cool about rice from an ionomic processing of matter kind of perspective is the fact that rice is grown in a tremendous diversity of environments, right? Everywhere from the rain-fed lowlands of Southeast Asia to the elevated hillsides of West Africa to the irrigated deserts of North America. And all of these different soil chemistries and all of these different locations across the world present unique and wonderful opportunities for rice and also challenges for rice in terms of acquiring proper nutrition and keeping out all of the toxic elements. And so this is what we call Ionomics, and we'll define it a little bit further. But I really enjoy studying this phenotype because it has wide-ranging implications across a diversity of the human condition. Everything from nutrient inputs, both on the terms of the plant and on the terms of the human, global climate change, human health and wellness, and abiotic stress tolerance. These are not minor global challenges, and Ionomics as a phenotype interfaces with each one of them in different ways. So what is Ionomics really? I've said for 3 years now, and I still maintain, that Ionomics is an invented term designed to invoke feelings of post-genomic era novelty regarding a decades-old technology. But more functionally, Ionomics is the mineral, nutrient, and trace element composition of an organism. It was the term was coined by a man named David Salt, and the irony of his last name is not lost on me. But functionally, what we want to try to accomplish with Ionomics, at least with this project, in the parlance of the athletic contest, is that we want to know what positions are there on the field and how do those positions interact in order to score a touchdown, right? And more importantly, and perhaps more interestingly, how does it change the outcome of the game when we make a player substitution, right? If you take out Tom Brady and put in Tim Tebow, does that change how you're able to score a touchdown? So we're going to look a little bit at the functional genomics of mineral nutrition. So I walked into Cornell and I stumbled into this amazing collaboration that has uniquely empowered me to be able to answer the very questions that this poses. Susan here is my advisor and provides a lot of germplasm and genotyping expertise. Leon Cochin on my committee has been invaluable in helping me with high throughput phenotyping. Jason Measy, who's not on my committee, has been super helpful in terms of bioinformatics and statistical support in associating genotype with phenotype. So I want to take just a couple of minutes really briefly to go over some of the tools, not all of the tools, but some of the tools that are available in these different spheres, because I'm going to refer back to those tools later as we've used them. So here, this is Susan, and this is the rice community at large. And the rice community at large and Susan have all provided tools that we've been able to take advantage of. The first of which, and probably most importantly, is RDP1. Rice Diversity Panel 1 is a panel of rice varieties collected from 79 countries. They represent all 5 subpopulations of domesticated rice, and they've been genotyped with a high density, what we're calling the high density rice array, which is a high density Affymetric SNP chip, which has been developed for rice and has been done on this panel. We also have 125 lines that have been put together by the Rice SNP Consortium that represent germplasm from both public and private sources, and these lines have all been fully resequenced. So we're going to take advantage of some of those resequenced lines as well. We have numerous biparental populations which have all been GBS, is that a verb these days? Have all been genotyped with either GBS or the high density rice array. And then this is where the rice community comes in. We also have a fully annotated, stable, and queryable genome assembly, which has been, our reference genome is Nipombare, which is a temperate japonica cultivar. And this has also been tremendously useful. Leon, for his part, has contributed sophisticated molecular physiology expertise, decades of hydroponics experience, reliable growth chambers in controlled environments, as well as powerful analytical chemistry, mostly in the form of Mike Rutsky, who is now the director of the Cornell Nutrient Analysis Lab that can be found here on the 7th floor of Bradfield. Jason has worked with his team and with Susan to develop statistical pipelines that allow us to do genome-wide association mapping using mixed models. What's been really nice is that he doesn't just give you a text file with a bunch of SNPs and p-values, but then he's also been able to produce powerful visualizations of those GWAS results. Tony Greenberg here began in Jason's group, and I think now is more squarely under Susan's umbrella, has been invaluable in his assistance with Bayesian statistical modeling of our phenotypic covariates. We have gene co-expression databases that are available, and we also have comparative genomics resources that are available. So those are all of the tools that I sort of found in my sandbox as I walked into this tripartite collaboration between Jason, Susan, and Leon. So these are some of the questions that we want to answer using some of those tools. What is the relationship between phenotyping in controlled environments versus field environments, and what advantages or disadvantages does it offer for studying ionomics? What does multivariate analysis tell us about relationships within this correlated network of phenotypes that we call the ionome? And how can genome-wide association analysis be used as a tool for both for cultivar enhancement as well as plant genetics? And what other tools do we need in hand in order to leverage its power? So number one, for a long time, and I think really only in the tenure of my time here at Cornell has this changed, but for a long time genotyping was the primary constraint to genetic analysis. But for a lot of crops, and rice in particular, the king is dead. And phenotyping has become the major bottleneck, right? And I don't think that phenotyping is going to be anywhere near as a benevolent dictator as genotyping was. So I think we're in for a real challenge. Because there's all sorts of different considerations you need to think about when you're trying to approach a phenotyping experiment, not the least of which is how you are going to collect that phenotype data. Are you going to use field environments and sample across multi-locations? Are you going to use controlled environments? And then once you've got that, you need to think about how you're collecting your data, how you're going to store your data, how you're going to adjust your data until eventually you can associate it with genetic data, and then that brings you back up to germplasm again. So as we pondered this idea of mapping the rice ionome, we had to think a little bit about how we were going to phenotype these things and whether we were going to phenotype them under field or controlled conditions. They offer different advantages. The field conditions, most importantly, I think, maximize relevance to breeders and farmers and allow you to estimate GBI effects. But controlled conditions also allow us to minimize environmental variation, and for particularly ornery phenotypes like the ionome, this can be really valuable. It turns out David Salt, once again, is independently funded by the NSF to work with the exact same germplasm that we worked with, but to grow it in multi-locations across the world and look at the grain ionome. So alongside of his project, we developed a project where we decided to look at, if he's looking at grain, we wanted to look at roots and look at shoots, and we were going to do that under controlled conditions, right? So the only thing that would make this better is if we were studying corn, right? Can you imagine? Salt and cob, ionomic variation in domesticated maize. But alas, the opportunity is lost. But anyway, so there we have it. We settled on a controlled condition, and we used resources in Leon's lab to be able to accomplish that. Hydroponics was our platform of choice. Hydroponics allowed us to survey all the elements of biological interest to rice, all the mineral elements, at one time, right? So you're going to be hard pressed to find a soil that'll have everything that we stuffed into this hydroponic nutrient solution. It permits the analysis of roots as well as shoots, and it provides a stable and controlled environment which helps us to increase heritability. In order to approach this, we had to design a nutrient solution. So we wanted to design a solution that provided all of the nutrients that rice needed to survive and do well. Some beneficial nutrients that you don't necessarily need in a hydroponics nutrient solution, but we're interested in seeing variation for uptake, as well as some crazy toxic things that people don't normally include in a nutrient solution, but we also wanted to include in our analysis in order to make it ionomically comprehensive. All told, we have about 24 elements in the analysis that we're looking at. I won't belabor this point, but this is basically the pipeline that it takes to take a plant from the hydroponic solution and turn it into a data point on an ICP spreadsheet. And there are several points in this pipeline where we've been able to enhance the precision and accuracy of our phenotyping platform. I'll mention briefly that we grew the plants to be 6 weeks old, which gave us almost 200 milligrams dry weight root tissue. And that was a substantial improvement from what's typically been done in the past. We added a rare earth element called yttrium to each sample, and that allowed us to calculate a very specific dilution factor for each individual sample rather than making an assumption that everything was diluted equally. And then Dr. Rutsky here, who runs this ICP, not only purchased this brand new ICP 6 months early so that we could do this, but Mike also used data and samples from our project to develop methods on the ICP. So the ICP was also fine-tuned to detect the elements in the range of concentrations that we were going to see in our samples. So that was also really helpful. And as you can see, one of the advantages of all of that was that we were able to increase our accuracy and precision. So this is our % relative standard deviation for the measurements in each of these in red, and in green is our accuracy, which is the lower detection limit of the machine. Okay, so just to give you an idea, strontium here, we can detect strontium up to 0.8 parts per billion. So that's a fairly low number. To give you an idea of how good these are, we'll show you that in the Arabidopsis community, they have published their % RSDs for what they were doing with their Arabidopsis mutants, and this is the fold difference here. So you can see that for many elements, we can detect them many-fold times more precisely than they could. Largely, that's due to those things that I just mentioned, the fact that we're analyzing a lot more tissue than they were, and that we've taken measures to ensure that we're not introducing variation through the pipeline. Tony Brilliant Greenberg has also then takes this raw data that has been produced off of the ICP and organized, and he creates a multivariate hierarchical model and produces a Bayesian estimate of line means, right? And he does this by nesting replicates within lines, and lines are nested within subpopulations. I should mention two things. Number one, the experimental covariates as well, things like which tub was the plant grown in, where when that tub was the plant grown, who harvested the tissue. All of those things are taken into account simultaneously with all of the other phenotypes. So this illustrates just one phenotype, but he's looking at all of the ionomic phenotypes at the same time. So these means are actually vectors and the variances are covariance matrices because he's doing everything all at the same time. His statistical analysis combined with the precision of our phenotyping platform has lent itself to reasonably high heritabilities. And those that have really low heritability actually correspond perfectly with those elements for which we have very low precision to start with because they're just difficult to measure in the ICP. So what does multivariate analysis tell us about relationships in this correlated network? So this is a PCA biplot of the shoot ionome in our analysis. And what it is meant to do is illustrate relationships between phenotypic covariates in this network. And what we can see from this plot is simply is that there are relationships. Those relationships differ between the different phenotypes. And interestingly, this one down here is dry weight biomass of the shoot, right? And then just to give you a hand with interpreting this, if you're unfamiliar, anything that shares a perpendicular orientation here has no correlative relationship. Anything that shares a bidirectional relationship is negatively correlated. And two things like molybdenum and calcium here are positively correlated. Now, this is only interesting in and as far as this is interesting. It turns out that the relationships of the shoot ionome change as you move across genetic subpopulations, right? Now, this is kind of busy to analyze, so I'm just going to highlight one token example here. Here is biomass dry weight, and here is cadmium. In indica and in temperate japonica, the two are reasonably negatively correlated with one another, okay? In Ausch and tropical japonica, they're still slightly negatively correlated, but they share a much more perpendicular relationship with one another. So think about this in terms of the Green Revolution, right? In the 60s and 70s, semi-dwarf genes went viral before going viral was really cool. And what ended up happening was all of the wheat and the rice shrunk in terms of dry weight biomass of the shoot tissue. We might hypothesize, based on this, that also led to an increase in cadmium, okay? So it turns out that if you look at these correlations, the indica and temperate japonica germplasm has a very strong and significant negative correlation with cadmium and dry weight. Ausch and tropical japonica do not. So if it turns out that this hypothesis holds water, right, that cadmium concentrations went up as biomass went down, Ausch and tropical japonica might be good sources of germplasm to use in order to try and ameliorate that effect. Yeah?
This is all in the shoot as opposed to in the grain?
This is all in the shoot, okay? This is not in the grain. So in terms of that relationship, the two things that's important to remember is, number one, just because it's in the shoot doesn't mean it gets to the grain, okay? That's important to know. But number two, it's not ever going to get to the grain unless it goes into the shoot first. Okay, so those are two important physiological principles to keep in mind. Okay, so this is where we start getting into some of the more fun stuff with the genome-wide association mapping. How can GWA analysis be used as a tool for both cultivar enhancement as well as plant genetics? Okay, RDP1, plant breeding department. Plant breeding department, RDP1. Okay, 500 lines, both wild and domestic. We're going to show you the analysis of the domestic lines. There's 400 domestic lines collected from 79 countries. They've all been genotyped with a high-density SNP array. And they've all been grown in hydroponic conditions. If you look across our whole genome-wide association analysis, you'll find that we had 362 hits. This is keeping in mind for 50 phenotypes. 24 elements plus dry weight on roots and again on shoots. Between those, we had 362 hits. The average hit length was about 1.5 cm. In rice, 1 cm is equivalent to about 240 kb, which has about 25 genes. What's interesting is our resolution tends to be fairly high. 97% of our hits are less than 5 centimorgans, which is fairly good considering if this was a biparental Qtl mapping population, we might be looking at a Qtl that's sitting on 15 or 20 centimorgans, or I've seen Qtl that take up entire chromosomes. So the resolution is good. If we look at hotspots in the genome, so these would be Ionomic hotspots, where several traits have indicated the same region as significant. There's 10 of them in the genome. I'll highlight 2. This one's really interesting because it's a hotspot for root traits, but only when you consider the temperate japonica germplasm. If you include everything else, these hits aren't there. The second one is similar to it, except it's only for root traits in indica. Okay? So it's clear once again that the genetic architecture of complex trait variation in these different subpopulations is often different. You might hypothesize that in these regions, there could be genes that serve as master regulators of ionomic homeostasis. And if that indeed proves true, perhaps those regulators also differ across subpopulations, which I think is really interesting. Another interesting correlative relationship to think about is where we intersect with the Qtl mapping. So Qtl mapping is important because it validates GWAS peaks. It's important because Norton et al. 2009, who surveyed the leaf and grain ionome under field conditions, this is their Qtl in and as far as they intersect with the GWAS peaks that we saw. And I'll highlight one example here is a cadmium Qtl for cadmium grain concentration in the field, and it co-localizes with a cadmium shoot concentration in our analysis as well. So some interesting relationships as you try to intersect this data set with other data sets that have been collected. We have a lot of GWAS. Like I said, 24 elements plus dry weight times 2 tissues times 5 subpopulations means 250 Manhattan plots. And in order to develop some kind of port of entry into annotating some of these GWAS peaks, we embarked on a candidate gene analysis. Okay? So using gene ontology and comparative genomics, we were able to identify about 1,200 candidate genes in rice that are putatively associated with ionomic variation based on their electronic annotation in the database. We have 362 peaks in our GWAS analysis. Oh, and I should mention that 28 of these genes are cloned and characterized in rice, so we know and have verified their function. The intersection between these, we had 131 of these candidate genes intersected with 163 of our peaks. 5 of those were cloned candidate genes. This is important to caveat that we don't really believe necessarily that these are all of the genes that are involved in mineral nutrition in rice. These are picked based on electronic annotation. Electronic annotation is fickle and somewhat circular in its logic. The other thing you want to consider is that RDP1 is small. RDP2 is coming, and it's going to be a lot bigger. It's going to be a lot harder to phenotype. But RDP1 is small, especially when you consider that we're mapping within a subpopulation. So we may have 400 lines, but if we're looking only at one subpopulation, oftentimes that sample size can go down to 50 or 60. Okay? So we also don't believe necessarily that we're finding all of the regions of the genome that are significant for ionomic uptake. And then thirdly, these 28 clone genes have all been cloned, not all of them, most of them have been cloned using mutants. And so there's no evidence necessarily that natural variation of these genes is also controlling ionomic variation. Okay, back to the GWAS. Now we have a port of entry into annotating peaks. We have 250 plots, and I'm going to tell you about 2 of them. If you have your favorite element, come talk to me. We can go and look at the data. But for now, we're going to talk about 2. So now I want to come back to Bangladesh. Okay, Dr. Kaufman says that there's 140 million people that live in a land area that's about the size of Iowa. Okay, that's incredibly densely populated. And they get 76% of their caloric intake comes from rice. If we get 2 more feet in sea level rise, the entire rice growing region of Bangladesh is going to be underwater. In the meantime, seawater intrusion is creating tremendous saline and sodic soil problems for the rice growing regions of Bangladesh, so much so that it's referred to as the white plague. Okay? So sodium is an element that needs no introduction. I should point out that Bangladesh is a token example, but that salt problems are worldwide. Largely, they're along coastal areas where you get seawater intrusion, but also in the arid areas because of irrigation. Okay? So as you irrigate, you exacerbate salt problems in soil. So to give you a little bit of an idea of the topography of sodium in our analysis, you'll see that it's normally distributed across the subpops, and I want to point out that Ausch here typically accumulates less sodium in the shoots than most of the other subpopulations. That's important because Ausch has also been the donor for most of our significant alleles for salt tolerance and other abiotic stresses. I also want to point out that this data was collected under very low ambient non-toxic sodium conditions. This is in our hydroponics nutrient solution. So this is not necessarily under salt stress. Here is our GWAS across all these different subpopulations for sodium concentration in the shoot. And you can see, particularly in Ausch, we see some really significant peaks. There's here on 1, there's one here on 1, here on 4, and another one here on 8. These peaks co-localize with important and well-known and characterized sodium transporters in the rice genome. So there's the HKT family of high-affinity potassium transporters is well known to transport sodium. There's 7 of them in the rice genome, and we're hitting 5. So that's pretty cool. But in addition to that, we're hitting other things too that you would not have necessarily implicated in controlling sodium shoot concentration. So let's zoom in. This is just the Ausch plot, and we're going to zoom in on these 4 chromosomes where we see our most significant hits. Here on 1 and 4, here's our big one on 4. It's sitting on top of 3 known sodium transporters. Here's 2 on 1. The Saltol QTL is a QTL that was discovered many years ago now that Erie is working as well as others to find map and to integrate into Bangladeshi varieties. It's right here on chromosome 1. Here's another, right here on chromosome 1, another HKT transporter. In addition to these loci, we have several loci where we have significant hits, and really interesting and believable candidate genes. So if you wanted to come up and give me the most generic definition of a sodium transporter, I would say a monovalent cation hydrogen antiporter. And we have several of these candidate genes that are annotated as such underneath big significant peaks. The one that we're going to focus on is this one right here, the SLT1 gene in rice. It happens to be sitting underneath this peak here, and we'll talk a little bit more about that. There's also several other peaks on each of these chromosomes for which there are no obvious candidates underneath, and we don't really have a port of entry into understanding what's going on here without doing some more genetic analysis. So SLT1, sodium lithium tolerant 1, okay? It's a small heat shock protein, and it turns out it's a unique class of small heat shock proteins that's unique to plants. So animals don't have this kind of heat shock protein. They took it out of tobacco and out of Arabidopsis and threw it into yeast. Oh man, my yeast is cut off. That's okay. You'll believe me. They threw it into yeast, and it turns out it made salt-susceptible yeast tolerant to sodium and tolerant to lithium, hence the name, SLT1. It turns out that they also took it out of rice and threw it into E. coli. So the guys that took it out of rice, they're heat shock guys. So they took it out of rice, threw it in E. coli, and it turns out it lends thermostability to E. coli as well. And it has a well-characterized role as a protein chaperone. Could we spend 2 or 3 years fine mapping this thing, verifying that SLT1's causing this significance, and really defining what SLT1 is and does? Yes. Does Bangladesh have time for us to do that? No. So we decided to take a different approach to characterizing this. So here's our hit. Here's our phenotyping platform. The question we want to ask is, does this mean anything under salt stress? Because this hit was discovered under ambient sodium conditions. So this is TGFI. Thank goodness for Ise. So Ise is a former student in our lab. She has been working on basically fine mapping and cloning these yield QTL that resulted from a cross between an Ausch-like wild rice variety that we lovingly call IRGC 105491. And it's been crossed with a US adapted tropical japonica variety known as Jefferson. And so Ise developed all these cool families, and we found one of them. Turns out it's heterozygous right in this location on chromosome 1, which we're really interested in investigating. And it's mostly clean, but it does have some integration still in the background because it's been back-crossed several times to the recurrent parent. So Ise had also selfed this line, and we were able to pull out lines that have interesting and divergent genotypes at the point in chromosome 1 that is interesting for our analysis. These are very similar to the heterogeneous inbred families that Jackie presented on last week, right? I prefer to call them mills. They're mostly isogenic lines, right? So mill 1 here has the Rufi allele, okay? So this is the Ausch-like rufopogon allele, which we expect to be the salt-tolerant one at our SLT1 locus. And mill 2 here has the Jefferson allele right there. So comparisons between mill 1 and mill 2 are comparisons of the effect of that locus. Both of them have Ausch-like alleles at this other locus next to our locus, where we have the saltol QTLs located and these sodium transporters are located. So we're basically surveying the effect of this region in a salt-tolerant background, in and as far as this thing is salt-tolerant. And then mill 3 here is just a control for the background. So where mill 3 differs from Jefferson, that's gives you an indication if the genetic background has any real impact. So we grew these things in a growth chamber, and we stressed them out with all sorts of salt. So here's our controls, and here's our treatments. I should mention, too, this is dated yesterday. So wild parent spawned super salt tolerant rice. This comes from Erie. This is not the same wild parent, but the point is the wild varieties of rice are very interesting in terms of their phenotypic value if you can identify the integration that you need to bring over. So let's have a brief look at sodium shoot concentration. Here's our Ausch-like wild variety of rice. Here's tropical japonica. These are the two parents. They're not statistically distinguishable from each other, but there's a notable drop in sodium concentration in the shoots of our wild parent. If you compare mill 1 and mill 2, which are the two varieties that test the effect of our region, however, we get significant difference. Mill 2 here, which is Ausch-like at our sodium, our region that we know is important for sodium, is really very similar to the parent, Jefferson. And then we see here that mill 1, which has our Ausch-like allele at the SLT1 region that we're investigating, is much more similar to rufopogon. If you look at sodium-potassium ratios in the chute, which is a well-known metric and proxy for yield under salt stress, a similar pattern emerges. So we conclude that be it SLT1 or be it something else, underneath this peak, we're seeing that there's a significant effect for sodium chute concentration, and it warrants further investigation. Quickly, we're going to zoom through molybdenum. Okay, so that's the sodium story, and the sodium story is a story about germplasm and lines and phenotypic effects and starving people and salty soil in Bangladesh. Molybdenum is much less dramatic, but it's a really cool element nonetheless, especially genetically. So molybdenum is important in rice and in all plant life for 4 enzymes in the whole plant. And so a lot of people think that maybe it's not so super significant. And it's not, unless you care about things like nitrogen and acid rain. So nitrate reductase requires molybdenum, and it's the rate-limiting step in nitrate assimilation. Sulfite oxidase is an enzyme that requires molybdenum and is the primary enzyme for detoxifying sulfite, which is acid rain. Aldehyde oxidase is the final step in ABA biosynthesis. Okay? It also requires molybdenum. So molybdenum is important in as far as it affects all of these different things. Again, to give you an idea of what molybdenum looks like in our population, Ausch tends to accumulate more molybdenum in the shoots. Everything else tends to be lower. Indica especially tends to be lower. Okay, so pay attention to that. So Jason Measy, who is a statistical genomicist and typically works in the human community when he does this, I remember the day that he comes to me and he says, "Josh, tell me about molybdenum." Oh, except he couldn't pronounce it, yeah. But, you know, tell me about molybdenum. And it turns out that he was really excited about it because there's this tremendous peak right on the top of chromosome 8, the likes of which we've never seen before in GWAS and rice. 10 to the minus 15 is super significant, right? It turns out that this peak here co-localizes with a gene annotated as a putative sulfate transporter. Sulfate, molybdenum, and molybdate are chemical analogs of one another. And that gene shares 59% sequence identity with a similar molybdate transporter recently cloned by the other Ionomics group in Arabidopsis. Okay, so we felt pretty confident that we're onto something. You'll notice there's a hit in indica, a small one. There's no hit in Ausch. There's no hit in temperate japonica, but there's a significant hit in tropical. And you can see that tropical japonica has a lot of within subpop variation. So we're clearly segregating in that subpop. So what we wanted to do is look at haplotype analysis at this MOT1 gene in RDP1. So this is the high density rice array genotypes in RDP1. Here's our gene in the black box. The blue is the 5' UTR. The red is the only SNP we have on the array that's in the exon. There's only one exon. And the other blue is in the 3' UTR. Okay? And we're in this teeny tiny little LD block, which is also really significant and perhaps explains why we're seeing that there's peaks in some and not in others. If you look, yeah, these haplotypes explain some of the phenotypic variation, but that's not surprising because the haplotypes also segregate with subpops. So haplotype 2 is all mostly temperates, and then haplotype 1 is mostly indicas and tropicals, right? So we're just basically tagging the variation that's happening across subpopulations. But if you look within a subpopulation, things start to get a little bit more interesting. So here is the same haplotype. Here's our gene. I didn't mention this is the 2 SNPs in our array that are upstream of our gene. So these are in the promoter region. We have 2 haplotypes of appreciable frequency in tropical japonica, and they explain a significant amount of the phenotypic variation in tropical japonica. And they differ by this SNP here in the 3' UTR and this SNP here in the promoter region. So if we take the exon and we expand it out using our 125 lines, 50 of which overlap with RDP1, so we have phenotype data for them, and we look at the sequence of the exon, we see that the exon itself, which is in red here, is completely monomorphic in tropical japonica. And while we see significant differences in molybdenum, those differences are tagged by these 3 SNPs in the 3' UTR. If we expand the promoter, similarly, we see much more divergent variation. We have wildly different promoters in these 2 haplotypes, which are the 2 that characterize tropical japonica. And we see that these 2 haplotypes explain a significant amount of the phenotypic variation in the population that we have phenotype data for. qRT-PCR is underway, but the results are ambiguous. Okay, so you can imagine how much of a bump in gene expression you need to increase your molybdenum from 3 ppm to 4 ppm, right? So we'll notice that these 2, we've looked at these and we notice that these 2 are expressed in very similar ways, and we need to increase the resolution, their ability to discriminate between things that are expressed at low levels. Okay, so this is another cool story. Okay, indica, really low for molybdenum. Turns out that guy right there has twice as much molybdenum as any other indica in the whole analysis. His name is Sigadis, and he's an Indonesian variety. Maybe it's a she, I don't know. Turns out, if you look at the haplotype of this character right here, it's the only indica that has an Ausch-like haplotype at MOT1, okay? So this haplotype is common in Ausch, it's rare in indica, and this particular line has twice as much molybdenum as everything else. So more evidence that says MOT1's doing something. This is also really cool, okay? So the Japanese have put together this tool called RiceFriend. And RiceFriend is a gene co-expression network database where you can basically take a gene, throw it in their database, and it'll pull out the gene co-expression network associated with that gene. So we put MOT1 in there, and we get this cute little gene co-expression network with 11 nodes and 10 edges. And it turns out, if you look there, this gene here and this gene here, which are in the co-expression network of MOT1, co-localize with hits for molybdenum root concentration in temperate japonica. And since all of this data is based on Nippon-bore, which is a temperate japonica, that's not terribly surprising. At least that we're specific to temperate japonica. But it's really cool that we're co-localizing with hits. So, questions at hand. What is the relationship between controlled environments and field environments? Well, our controlled environment has given us phenotypic precision, breadth, and uniformity. Breadth in terms of elements, because we can't find a soil that contains all of the elements we're interested in one time. And also in terms of lines. All of our lines aren't going to grow well nor flower in all these different locations across the world because they're surveyed from all these different locations across the world. So this gives us phenotypic breadth and phenotypic uniformity, and is powerful when used in combination with targeted germplasm and hopefully with field phenotyping as we get David's data. What does multivariate analysis tell us? It tells us that there are relationships between the ionome, which is a correlated network of phenotypes. Those relationships are important and driven holistically, not just necessarily by pleiotropy or drag. And they pose interesting hypotheses that we would want to test genetically. How can GWAS analysis be used, and what do we need to leverage its power? GWAS is a powerful, if still somewhat impotent, tool for genetic dissection. Okay? And we're continuing to improve it as time goes on. But in combination with other powerful tools like targeted germplasm, gene co-expression networks, and sequence analysis, it can lead to important discoveries. So what we see here is that this sort of thing is happening. Okay? As independent lines of evidence that you may not necessarily believe in and of themselves all converge on a single point, now all of a sudden we can develop powerful hypotheses about a single point and really drive an understanding of the genetic variation that underlies complex trait variation in rice. So to conclude here, because I've got 5 minutes left, which is perfect, I want to address the last question, the question I haven't posed yet, but the question you're all thinking because I made this really provocative title about building a better mousetrap. Ralph Waldo Emerson said, if you build a better mousetrap, the world will be a path to your door. And so I want to pontificate a little bit because this is my exit seminar and I'm allowed to do that. What is this better mousetrap, and how exactly do we maintain the comparative advantage? This concept has nothing to do with my project specifically and everything to do with our department generally. Okay? So we have all of these things at Cornell: challenging courses, spontaneous and exciting hallway conversations, well-attended seminars as evidenced by what we have here, interdisciplinary collaborations, international opportunities, support for Synapsis. The department is tremendously supportive of our graduate student and faculty group for plant breeding. Private sector involvement, the Pioneer Symposium, Symposia rather, is the latest example of that. And support for participations in national meetings. The department has always supported the grad students as we've endeavored to attend the National Association of Plant Breeders and other important meetings. Having these things, in my opinion, isn't the challenge, okay? Everybody has these things at some level or another. Nucleating these things is the challenge, okay? And I can tell you from raw, in the trenches, unedited graduate student experience that the nucleation of these things is where education happens. And the nucleation of these things is where creativity happens. And the nucleation of these things is where innovation and imagination takes flight. And it takes a graduate student, not necessarily me, but all of the graduate students in here, to bring this nucleation to a head, okay? That's part of the synergistic role as we have as mentors and mentees in our department. And so I would encourage you, I don't know, I'm leaving. The first week of June, I'm in Iowa. First week of June next year, I'm in India, and I'm rocking it, right? I hope to maintain collaborations and relationships with everybody in here, but I'm not going to be intimately involved in the future of this department, at least not in the short term. So as you move forward, Whether we get to keep our name or we don't get to keep our name. As you consider the compromises that need to be made, consider this. Because for me, this is what I consider to be our comparative advantage. And if we lose this, then you've lost everything. You better cash in your chips because you've just turned in all your marbles. With that, I'm going to close, and I'm going to embarrass Susan.
She can't talk today. So.
And she can't refute it because she can't talk. I want to make a very special acknowledgement. Okay? Susan alluded to it in her introduction. And if you're interested, we can talk about it. But I grew up, proverbially speaking, on the wrong side of the train tracks. Okay? People that I grew up with, in the same circumstances in which I grew up, didn't attend Penn State. Most of them are attending the state Penn. Okay? Probabilistically speaking, I should not be here. I shouldn't be at Cornell. I shouldn't be delivering a seminar in the plant breeding and genetics department, and I should not have been able to nucleate all these powerful things we just talked about. But I have. And part of the reason why I've been able to do that is because throughout my life, strategically placed have been people that I can count on one hand that I refer to as enablers. They've simultaneously challenged me not only to elevate my vision, but also empowered me to accomplish it. In what has to be the most paradoxical emotion I've ever experienced, Susan, I'm simultaneously proud and deeply humbled to count you as both noble and great among the enablers of my life. And I think that we've gotten to know each other well enough that I think that you know exactly how much that means to me. With that, there's a cajillion other people that I need to thank. Not the least of which are people in Susan's lab, other graduate students, rotation students. They've come, they've gone, they've helped, they've left their mark in some way on this project and on myself. All of our collaborators in Arkansas who have been instrumental in the development of this panel and helpful in getting the genotyping and everything else underway. Leon's lab has been wonderful in helping me not only execute the phenotyping component of this project, but also interpret it in a biologically meaningful way, which I wouldn't have been able to do otherwise. And Jason's group is the one that's put it all together, that's helped me to visualize my own data and say, look, this is what it means. These are the relationships that are there, and these are the hypotheses that we can pose. I'm tremendously grateful for all of their work and how much of it has contributed to my own education at Cornell and as a PhD student. I've been funded by a bunch of different places. Cornell Diversity Fellowship, DuPont Pioneers provided funding. Leon is associated with the USDA and has provided a lot of funding for the phenotyping. And Susan's project with RDP1 and others is funded by the National Science Foundation. With that, I'll stop and take any questions that anybody has.